Hi everyone, welcome back to the Immunology series. Now in this lecture, we will begin a new unit focusing on immunological disorders. The first disorder that we are going to talk about is hypersensitivity. So let's get started. Here are the lecture objectives. We will begin the topic by briefly comparing and contrasting the 4 different types of hypersensitivity. Then we will look at how mast cells, Eosinophils, and Basophils play a role in hypersensitivity. And lastly, we'll explore how inhaled dermal and ingested allergens cause type 1 hypersensitivities. So the whole lecture is focusing on type 1 hypersensitivities in greater detail. Protective functions of the immune system rely on recognition of self from non-self and recognition of pathogen molecules as foreign. Now, however, besides infectious agents, humans come into daily contact with numerous other molecules that are equally foreign but do not threaten health, and under normal circumstances should not trigger inflammation or adaptive immunity. However, when there is an unnecessary overreaction of the immune system to some environmental antigens or maybe self-antigens, it will lead to hypersensitivity reactions and so-called allergies. Now, many allergens actually are derived from plants and animals that we eat or present in the environment that we live and work and play. Now, it is believed that the prevalence of hypersensitivities is almost like an epidemic in industrialized countries and with high prevalence. However, the prevalence or incidence rate, it is difficult to assess. It is possible that the prevalence is underreporting because many people may never register on pharmacist or physician's radar, and because mild cases are often treated with over-the-counter medications. However, there are increasing number of people in the US and other industrialized countries have hypersensitivities, and it is believed it is the 6th leading cause of chronic illness in the US. Classically, hypersensitivity can be classified into 4 different types, and each one have slightly different effector mechanisms. And let's first look at type 1. Now, type 1 hypersensitivity is an immediate action, occurs immediately upon exposure to an allergen. It is the most common type, and it is also known as allergy. Now, type 1 is mediated through actions of IgE and mast cells by the most part, and is most often caused by inhaling particulates such as plant pollen. The reaction severity is highly variable, ranging from runny nose to possible anaphylactic shocks. Type 2 hypersensitivity is caused by small molecules covalently bound to cell surface components of human cells, producing a modified structure perceived as foreign by the adaptive immune system. And B cells and antibodies are all involved in this type of hypersensitivity. Cytotoxicity is caused by IgG, complement, and phagocytosis. For example, penicillin allergy is a common example of this type of reaction. Again, the severity can range from hives, rash, to anaphylactic shock. Type 3 is triggered by the formation of small soluble immune system complexes of IgG plus allergen plus the complement plus red blood cells. Now, precipitations of the immune complex in tissues, usually in small blood vessels or alveoli, will initiate further complement activation and inflammation. For example, non-human proteins, such as that with serum therapy, can trigger type 3 hypersensitivity. Type 4 hypersensitivity is triggered by overreactive T cells. Now, the reaction is delayed and is typically developed within 1 to 3 days after exposure. Now, for example, an allergen can chelate with human protein and be taken up by antigen-presenting cell and processed and presented to T helper cells. This type of hypersensitivity is also associated with some autoimmune diseases and organ graft rejection. And examples of that of type 4 would be IV poison, poison IV reactions or nickel reactions. I don't know if you have noticed the common theme yet, but let's take a look here. The first three types of hypersensitivity reactions all involves the actions of antibodies. Type 1 involves IgE, and type 2 and 3 involves IgG. But in contrast, type 4 is unique that it is caused by effector T cells. This lecture, we focus on type 1 hypersensitivity because it is the most common and symptoms can range from mild sneezing to serious anaphylactic shock. Type 1 reactions are IgE-mediated. IgE is uniquely among the immunoglobulins found in peripheral tissues. Like any other antibody, it is synthesized by plasma cells, which was activated by Th2 cells. Now, in allergic reactions, the synthesis of IgE is favored above the synthesis of other antibody isotypes. And inhalation type 1 hypersensitivities occurs in much the same way as inhalation of any other acquired immune response, for example, antigen that is presenting. And it is noted that IL-4 drives isotype switching to IgE in this case. Now let's look at the details of how type 1 is driven by IgE. The Fc region, or the constant region of IgE, is the portion of the antibodies that bind to the cells, which binds to special receptors on the surface of mast cells, for example. Now these special receptors, we have a name, is Fc epsilon receptor, which has a high affinity for IgE. Now, these receptors are expressed constitutively on mast cells and basophils, meaning they are always there, and are inducible on eosinophils. It is very unique that this type of IgE and receptor binding stays essentially irreversibly bound to the effector cell surface, which is not a feature shared by other antibody types. During sensitization, allergen is cleared from the system, and all the excess antigen-specific IgE remain bound by the Fc region of the receptor on the cells. And these stable complexes provide these cells with antigen-specific primed IgE and Fc receptors that are ready and waiting to bind to more antigen or allergens. So we use mast cells as an example, but in fact mast cells, basophils, or eosinophils, they all are capable of binding to the Fc region of IgE. And these cells are packed with granules containing especially histamine, but a variety of inflammatory mediators. Now, in those mediators, some are preformed, meaning they can be released upon first degranulation. At the same time, transcriptions and translations happens and to synthesize inducible mediators of inflammation. Now, when the IgE and the Fc receptor complex on the mast cell surface encounters allergens a second time, it will quickly release of granule contents. We call this process degranulation. Now, the process continues and more inflammatory mediators are synthesized. So in the last slide, we mentioned mast cells, eosinophil, basophils. Now let's look at each cell type more specifically. The mast cells reside in mucosal and epithelial tissues lining the body surface, present in all vascularized tissues except the CNS and retina. Now, they serve to maintain tissue integrity and alert the immune system to local trauma and infection and facilitate the repair of damage. During the IgE-mediated degranulation, histamine, heparin, and TNF-alpha are released, Animal oxygenase and cyclooxygenase are also involved to generate leukotrienes and prostaglandins. Now, all of these inflammatory mediators can increase vascular permeability and recruit more immune cells to the location. Moving on to eosinophils. Remember, we're still talking about type 1 reactions. The eosinophils are similar to mast cells, and they are specialized granulocytes. They can release toxic contents in response to IgE binding, and they can perform killing and also induce or amplify inflammation. They reside in connective tissues underlying epithelia, respiratory, gastrointestinal, and urogenital tracts. They are there to secrete pro-inflammatory molecules and toxic proteins to kill parasitic worms. But these toxic proteins can also damage host tissues. Notice that Th2 cells release IL-5 to promote eosinophils to proliferate and following an infection. IgE also mediates eosinophil degranulation and secretion of histamine and reactive oxygen species. We have seen that a high amount of eosinophils in the blood can cause high amounts of tissue damage. In fact, chronic asthma is characterized by an increased presence of Th2 cells, eosinophils, neutrophils, and other leukocytes, and eosinophils are the principal cause of airway damage that occurs with chronic asthma. Moving on to basophils. Now, the granules of basophils are similar but not identical. Their content are more similar to mast cells. Now, although they resembling mast cells in some way, they are developmentally more closely related to eosinophils. They have a common stem cell precursor. They are also active against parasites, and IL-5 stimulates their synthesis. Basophils normally constitute less than 1% of the white blood cells, which for decades made them more difficult to study than other leukocytes. And during an innate immune response, basophils are recruited to infected tissues where they are activated through their toll-like receptors and other receptors of innate immunity. In the infected tissue, basophils contribute effector functions similar to those of eosinophils. Now, at the beginning of the adaptive immune response, basophils are also recruited to the secondary lymphoid tissues, where their secretions of IL-4 and IL-13 initiate the polarizations of antigen-stimulated T cell toward a T2H2 response. Here is a very nice graphical summary that I pulled from a review article that was published in Nature Review Immunology back in 2007. It shows mast cells, basophils, and eosinophils play a role in type 1 hypersensitivity. In fact, the cellular and chemical mediators of allergy are the same branch of the immune response that is engaged when the body is attempting to prevent colonization or infection by parasites. Now, in fact, parasites are multicellular organisms, and if you look at it that way, essentially they are tiny small animals. And when we think about our immune response so far, we've talked about, having cellular response, having phagocytes and all these, and components from these immune response are, although effective against viruses, bacteria, or even fungal pathogens, which are relatively small, they are ineffective against parasites because they are relatively big compared to these other microorganisms. So, and as parasites penetrate or adhere to our tissues, it would damage our cells and release small proteins and peptides that is causing symptoms. And what happens is that the symptoms or the reactions that is produced by histamine and other leukocytes or leukotrienes can physically expel the worm, and therefore there's that overlap of allergy response and parasitic defense. Now, between allergic reactions and parasitic infection, there are some very interesting links that people have observed. Now, allergies is well known and prevalent in industrialized countries, but it is actually not that prevalent in tropical regions. And in those regions, parasitic infections are considered an endemic, and in those peoples, they can cause increased Th2, IgE levels, and also increased eosinophils in the blood and increased mast cells in tissues. And we have observed that people with parasitic infections and high levels of IgE rarely develop allergic diseases. And you wonder why. Now, there's one observation that we've seen is that it is through a mechanism called competitive IgE. In fact, non-specific or allergen IgEs can compete with parasitic specific IgE for mast cells and basophils and eosinophil activations. And with the presence of a parasitic specific IgE, it can actually limit allergic IgE reactions or binding, and therefore, having less or oftentimes no allergy reactions. The physical effects of IgE-mediated mast cells degranulation vary with the tissue exposed to the allergen. Only those mast cells at the site of exposure degranulate, and once released, the preformed mediators are short-lived. Their effects are therefore localized. For example, IgE increases violent muscle contraction to expel parasitic worms in the gut, and in the respiratory tract, IgE induces muscle contractions and secretions of mucus, and in the blood vessel, IgE increases blood flow and vascular permeability to promote lymph circulation. Now let's look at inhaled allergies. Well, we're still talking about type 1. Now this is a funny cartoon I got from the internet. The guy is holding a pistol, the female reproductive part of a flower that is full of pollens, at an allergy sufferers convention. Interesting. Let's first look at allergic rhinitis, or so-called hay fever. I don't know who doesn't have allergic rhinitis these days. I suffer heavily during the spring season particularly. All right, so in this case, it is still type 1, remember that. Now, antigens or allergens can diffuse across these nasal epitheliums after inhalations. And these actions are mediated initially by mast cells. Through activations and degranulations, it will change the blood vessel permeability and it will swell up and also increase mucus productions. And then afterward, this reaction is sustained by eosinophil recruitments, and they'll come into the site and activated eosinophils are shed into the nasal passages. Now, these symptoms, such as sneezing and runny nose, they're usually mild and self-limiting, and usually no long-lasting tissue damages. However, in chronic allergies such as asthma, it is a more severe chronic inflammation, and it is characterized by a shift from type 1 to type 4 hypersensitivity. Now, in such case, these allergens or antigens activate submucosal sensitized mast cells in the lower airway, so it's not only limited to the nose. And in those positions, there's also increased mucus, contract bronchial smooth muscle so that it will lead to narrow airways, and also increased vascular permeability and persistent infiltrations of leukocytes, including Th2 helper cells, neutrophils, and eosinophils. When there is a chronic recruitment of high levels of leukocyte in the lower airway and secreting all kinds of cytokines and chemokines, eventually there will be something called airway remodeling happening in the asthma. Now here is an example biopsy picture of how chronic inflammation in asthma remodel the airway in asthma patients. Notice that there's a lot of thickening of tissues compared to a normal biopsy. Now move on to dermal allergies. Dermal allergies usually associated with touching something, But another way to look at dermal allergy is that when clinical allergists try to detect or find out a person's sensitivity to a particular allergen, they will often inject a small quantity of allergens under the skin and to look at the reactions. And there are two types. The first type would be immediate reactions. Shown in the pictures, there is some, hives, a little bit bumpy, maybe associated with some itchiness. However, if the reaction is sustained and prolonged, there will be some late phase reactions and causing more widespread swelling or even some bumpiness that is filled of some fluid there. Now, it is through activations of mast cells in deeper subcutaneous or connective tissues. Another type of dermal allergy is a disease commonly called as eczema or atopic dermatitis. Now, the etiology is not well understood, but it is associated with a high level of IgE, and oftentimes they resolve with adolescence. Now, in eczema, it is characterized by itchy skin rash with some eruptions and sometimes have some discharge, and it is related to hay fever and type 1 hypersensitivities. What about food allergies? Food allergy, in another word, is ingested allergens, commonly caused by eating grains, nuts, some fruits, legumes such as peanuts, fish, shellfish, eggs, and milk. Now, in ingested allergens, these allergens are broken down into pieces of peptides by the stomach and intestine. And in fact, they can be a perfect source of peptide to bind to Th2. Now, however, it is relatively rare that IgE is present in specific for any type of these allergens. But when IgEs are present, it will trigger histamine release, and that will cause changes in blood vessel permeability and also smooth muscle contractions, leading to symptoms such as cramps, vomiting, and diarrhea. But regardless of the original site of exposure, these allergens can disseminate into bloodstreams and may cause even higher amount or systemic type of reactions, such as some of the itchiness, or may even cause difficulty breathing, and also in a severe case it may also cause anaphylactic shock. When we talk about anaphylactic shock, it is a systemic anaphylaxis. It is caused by allergens that reach the bloodstream and activate mast cells throughout the body. Now, typically, it can be introduced by a sting of an insect venom, or can even by food, such as severe peanut or nut allergies. Now, it can also cause by some drug injections as well. Now, it will cause these widespread of activations of mast cells, and you can imagine that the reactions or the symptoms will spread out into different major systems. And for example, in the vascular system, it will increase vascular permeability and causing some vasodilations and sharp drop in blood pressure. In the respiratory systems, it will cause constriction of bronchial smooth muscle, causing difficulties to breathe. And in the GI system, the epiglottis will swollen and further constrict airway. And of course, other symptoms such as stomach cramp, vomiting, diarrhea will still persist. And these conditions can be serious and lead to death. And generally speaking, death from systemic anaphylaxis are generally due to asphyxiations, meaning suffocated and cannot breathe because due to the constricted airways. So we just looked at how anaphylaxis can cause symptoms in the respiratory and in the vascular or cardiovascular systems, and these conditions can be fatal. And to rescue anaphylactic shock, we have a specific treatment, epinephrine, or under the brand name EpiPen. Now, it is an injection that is injected into the thigh, and it's indicated for anaphylactic shock, particularly to prevent a fall in the blood pressure and also reverse some of the bronchoconstriction pathway here. Now, if you are my pharmacy student, okay, you'll be familiar with these alpha and beta receptors. Through these alpha 1, it will mediate vascular smooth muscle contractions, okay, to bring up, to restore the blood pressure. And through beta 2, it would cause bronchodilation to reverse the bronchoconstriction process. And through beta 1, it would act as a positive inotrope or chronotrope effect on the heart, and to increase the blood pressure as well. With this slide, I'm concluding this lecture. My spring allergies are really acting up. Look, I am your father. Have you ever occurred to you that maybe Darth Vader were suffering from spring allergies? All right, I'll see you next time. Thank you.